If we're going to talk about pathophysiology, it makes sense that we would start with cell structure and function. So recall from high school, middle school, maybe elementary school, that eukaryotic cells are cells that have a nucleus. All animals, multicellular animals, have a nucleus and therefore are eukaryotic. So that's the type of cell that we're going to talk about. Just for reference sake, I have this slide from Guyton and Hall that illustrates the relative size of the cell. So a cell is 5 to 10 micrometers. For reference sake, 1 micron is about the size of a bacterium, and tiny viruses are 15 nanometers. So remember all these components. We're just going to focus on a couple of the important ones. Of course, the nucleus is the control center of the cell. It's the one that signals the cell when to do things, when to grow, when to mature, when to replicate, when to die, etc. It's also the location of the genetic information of the cell, that is the DNA. Now, the nucleolus is actually not a specific part of the cell. It doesn't have a membrane around it, but rather it is a concentration, an area of the nucleus that has concentrated RNA and ribosomal proteins. The ribosomal proteins are formed from the RNA producing proteins, and then they will go on to form ribosomes, which we'll talk about shortly. There are additional slides available that have the details of this, but I summarized it because, we're talking about the human body. We're not talking about cellular diseases. So I just wanted to quickly go through and remind you of what the cellular machinery consists of. So the endoplasmic reticulum is the part of the cell that synthesizes all the cellular and membrane components. There are two kinds of endoplasmic reticulum, which we just call ER. So the rough ER is the ER that has the ribosomes attached to it. And you can imagine that those are important in protein formation. So the rough ER is what produces proteins in the cell. The smooth ER is what forms lipids in the cell and, combines all the fatty acids, etc., to make the lipids. The Golgi apparatus looks very similar and, in fact, is closely related to the endoplasmic reticulum. But the job of the Golgi apparatus is to receive those transport vesicles that you see pictured here. These transport vesicles break off from the smooth or the rough endoplasmic reticulum. They get taken up by the Golgi apparatus, and then the Golgi apparatus processes them and makes all of these lysosomes and these secretory vesicles, which we'll talk about now. So lysosomes are formed, again, when they break off from the Golgi apparatus. And essentially, these are the components of the cell that are responsible for digesting cellular structures that have been damaged, or nutrients that are ingested by the cell, or unwanted junk like bacteria. They are essentially just bags of enzymes. And in particular, the lysosomes have the hydrolases. So a hydrolase is an enzyme that participates in hydrolysis. And they hydrolyze proteins to form amino acids. They hydrolyze glycogen to form glucose. And they hydrolyze lipids to form fatty acids and, of course, the glycerol. So what they do is they break down those bigger substances into smaller pieces that can be used by the body. Peroxisomes are similar but distinctly different. lysosomes have hydrolases. Peroxisomes have oxidases. And so the oxidases are present so that they react with these intracellular chemicals to make hydrogen peroxide. And if you remember from chemistry class in high school, peroxide is very reactive. And so the cell uses that hydrogen peroxide to oxidize substances that are poisonous to the cell. For instance, about 50% of alcohol that's ingested is oxidized by these peroxisomes. They also catabolize or break down long fatty acid chains. So that's the process. Those are the processes that are important to us inside the cells and should help you to understand when we shortly talk about metabolism inside the cell. But before we get to that, we have to talk about the thing that powers the cell, and that's the mitochondria. Now, I only learned this when I did some reading, that apparently each cell has hundreds or even several thousand mitochondria. I had no idea. You look in the textbook, there's a picture of one mitochondria in every cell, but that's incorrect. Because cells require so much energy to function, they have a number of these mitochondria. And what they do is they convert energy to forms that can be used in cellular reactions. And what is that form? We're going to talk about it all semester. ATP, right? That is the currency of the cell. And it's exchanged inside the cell for production of energy. Now, the makeup of them is less important, but it is important to know that a mitochondria are signaled by the nucleus to begin the process of apoptosis. So we'll come back to that when we talk about apoptosis. So let's talk about cellular metabolism. That's kind of the whole reason to talk about the cell, right? Because that's what our cells require in order to do their work. Whether it's cardiac myocytes that are contracting to make our heart beat, or whether it is the liver cells that are detoxifying things in our gut and in our blood, whatever. Cellular metabolism is how we get energy. So there are three different major food sources that we have. The macronutrients are protein, polysaccharides, which are carbohydrates, and fats. And so this illustration here just kind of abbreviates all of the mechanisms that are at work here. So let's start with sugars. Sugars go through this process called glycolysis. And we're going to talk about glycolysis in more detail, but essentially that produces pyruvate, which then goes to acetyl coenzyme A, which you remember enters the citric acid cycle, which you will have to memorize. No, I'm just kidding. I'm not going to make you memorize that again. It was bad enough in college, right? But what happens is that all of that produces energy. In addition to energy, we get water out of it, and then that gets combined with oxygen and gets excreted. Proteins are metabolized through hydrolysis again, where the amino acids are taken from them, and then the excess or the reactants then are ammonia and water, and then they get excreted. And then in fatty acid metabolism, the fatty acids release CO2, which gets excreted. So you get nothing for free in the cell. Everything has a price, and the price here is that we produce waste products that have to be disposed of. So let's look at each of these things in a little more detail and understand, starting with protein metabolism, that protein metabolism happens in the cell. Now, where does the cell get the protein from? Well, some of that protein is produced by the cell, right? That's what RNA does. But some of it is taken from the bloodstream. Well, how do you get protein out of the bloodstream? Well, there's a ton of protein that floats in the bloodstream. That's part of its job, right? Is to transport protein. And so the albumin that circulates in the blood is the most prominent, most prevalent of all the blood proteins. And what it does is it is in constant interchange with the cells to either add to their protein needs or accept the protein that is produced by those cells. And that maintains a constant equilibrium in the blood between the plasma proteins, the cellular proteins, and the amino acids. So all of those things are in constant equilibrium. By the way, we'll discuss this in more detail later, but when proteins are metabolized in the body, the remnant of that reaction is NH3. So that's ammonia. And you know that ammonia is bad for the brain, right? If you have somebody with hepatic encephalopathy, they have too much ammonia being exposed to the brain, and the brain stops working. So, or at least stops mentating, shall we say. So the way the body deals with this is it combines two ammonia molecules with CO2, and that produces urea. That's what urea is. It's just ammonia plus CO2. And that reaction produces urea with a remnant of water as a result. So that's another equilibrium that exists in our body. We'll talk about that more when we talk about the kidneys and liver function. Carbohydrate metabolism is the biggest component that we rely on for energy for our cells. Glycolysis is by far the most important step in glucose metabolism because it starts the whole process of producing pyruvate. And it's the pyruvate then that enters the citric acid cycle, though not immediately. So the first thing is that when glycolysis happens, it produces two pyruvate molecules and two ATP molecules. That's not very much, right? Remember that the Krebs cycle makes a whole bunch more, but we do get a little bit of energy out of the initial process of glycolysis. Then that pyruvate combines with the coenzyme A to form acetyl coenzyme A, and then acetyl coenzyme A gets into the citric acid cycle, combines with oxaloacetic acid and yada yada, a whole bunch of other steps that we don't really care about. But it enters into the Krebs cycle. The Krebs cycle then oxidizes these acetyl groups and forms a huge quantity of hydride ions. And at the end of this whole process, we end up with 38 ATPs for each pyruvate that enters the citric acid cycle. So that's a pretty good output, right? But what's required for this output? Don't ever forget that what we need for this output is oxygen, right? So this has to be done in an oxygen-rich environment. And if there's not oxygen, then your body uses different metabolic pathways to still be able to supply glucose and energy, but not in the same amounts. Oxidative phosphorylation is another method that your body can use. This is also aerobic, and it produces ATP by reaction of ADP plus a phosphate ion. In order to do this, it needs a catalyst called ATP synthetase, which is a protein that's in the mitochondria. So guess where most oxidative phosphorylation happens? In the mitochondria. So that's protein metabolism we talked about first. The second thing we talked about was the carbohydrate metabolism. Now we're going to talk about lipid metabolism. Lipid metabolism is a huge way that we get the acetyl coenzyme A that fuels the citric acid cycle. So this is one of the reasons that you need cholesterol in your body, right? You need fat in your diet in order to generate energy. So this is the way that it happens. We're going to talk more about this later in the semester, but essentially your gut absorbs these chylomicrons. They go into this process and fuel different levels of metabolic pathways, and we have all sorts of things to show for it at the end. We can measure ApoE, we can measure ApoB, we can measure adipose tissue. We know what the results of this have been. And of course, when we have somebody who's diabetic, that changes the metabolic pathway of lipid metabolism, not entirely, but it really makes a difference. And that's why we see the advanced coronary disease in diabetics. Again, we'll talk about that later, but just know that the metabolic pathway, the lipid metabolism pathway, is how we get acetyl coenzyme A that fuels the citric acid cycle. So we talked about all these other components of the cell, but we didn't talk about the plasma membrane. Well, I'm saving the best for last. The plasma membrane is really the magical part of the cell. And what's magical about it? Well, several things. One, it actually makes a barrier, right? And that seems sort of obvious, but that barrier is critically important because it keeps out the riffraff and keeps in the good guys. And by that, I mean that the plasma membrane can have a role in determining what gets in and what gets out. And that's really the crux of it all. The plasma membrane is responsible for getting nutrients across the membrane and getting rid of waste products through it. It's responsible for generating membrane potentials, that is, having a difference in electrical charge from the inside and the outside. And that becomes important when we talk about cardiac conduction, myocyte contraction, things like that. The plasma membrane has a role in cell recognition. So there are proteins on the cell membrane that allow your immune system to recognize self, and when that system works well, you are well and you're defending against disease. But when that system breaks down, you become autoimmune. You begin to destroy your own cells. Growth regulation is in part regulated by the plasma membrane. And then it's also critically important for sensing signals to respond to and adapt to changes in the environment. For instance, apoptosis comes from an extracellular signal, and your body then, or your cell, will respond to that apoptotic signal. So what is it that's responsible for this magical membrane? Well, it's really the construction of it. It's this lipid bilayer that exists, where there's a polarity to it that allows lipids to come across when invited, and allows water to also come across. That is chemically a very difficult thing to do. How can you have a polar compound have the same ability to access that bilayer or that membrane as a nonpolar compound? We all know that that oil and water don't mix, right? So if you have oil and you pour, you have water and you pour olive oil into it, the two separate. Well, that's what the lipid bilayer does. It acts for both sides and allows your body to respond accordingly. So from the start, the lipid bilayer has different affinities for different substances. Ions have no ability to permeate that lipid bilayer without permission, but certain things like CO2 and oxygen, some drugs, they have a high affinity. Now, I don't want you to get all freaked out here and worry that you have to memorize this. There's two important things that I want you to take from this slide. The first thing is I want you to see that there's different permeability that each of these substances have through that membrane. Water has a very high permeability. So water is able to easily go across that membrane. But things like the electrolytes have a very low permeability. It takes a lot to get those across. And in fact, we have to have a different method because the permeability is so low. Then there's things in the middle like urea and glycerol and glucose and things like that. Okay? High permeability for water, low permeability for electrolytes. On the other side, I want you to really take a close look at this because this is kind of the magic of how gradients exist across the cell membrane. You know that in the extracellular fluid, like serum, for instance, in the serum, the concentration of sodium is about 142 milliequivalents per liter, give or take, right? 135 to 145, we'll call it 142. And the potassium concentration is very low, right? It's on the order of 4 milliequivalents per liter. But look on the intracellular side. This is almost entirely opposite, where the concentration intracellularly of sodium is very low, about 10, and the intracellular concentration of potassium, on the other hand, is very high. So the intracellular and extracellular concentrations of these two electrolytes that we're looking at here are almost entirely opposite across the cell membrane. And that's why we have an electrical potential. So we'll talk about that as well. Now, a couple of things for you to remember. Diffusion is the movement of molecules in liquids or gases. All right. So you're going to want to remember that. And a couple of just basic science things that you should remember from high school are that there's an increase in the net rate of diffusion when the concentration difference exists. So we have a high concentration here, we have low concentration here, and so there's the net diffusion is a function of that difference in concentration. One way that we can affect that concentration is by changing the electrical potential. So when we have a net inward current of electricity, then the gradient can change. And that's what we're going to look at when we talk about how electrical potentials happen. The other thing that affects this is the hydrostatic pressure difference. So hydrostatic pressure is essentially blood pressure. Let's just think of it that way. When we have a higher hydrostatic pressure, that can facilitate movement across a membrane as well. Now, another term for you to remember is that osmosis is specialized diffusion of water. All right? And that results in homeostasis being achieved. And so in this example here, we have water without any electrolytes. So this is pure water, pure water. And this is a sodium chloride and water solution. What's going to happen here is that the osmotic pressure, the gradient between these two things, which is across this membrane, is going to force the water into this electrolyte solution to try to dilute the solution. Remember, it's the water that moves. And so this pure water is going to try to move over and dilute the sodium chloride solution in such a way that it will bring them to equilibrium. They'll be the same concentration on both sides. Well, you know that since there's no, since this is pure and there's no, since there's no sodium chloride on this side, all the water is going to go across here, right? It's just going to take all the water across. And that will never get to equilibrium, but the illustration is still about osmosis. And that is this movement of the water across a gradient to try to dilute this saltwater solution. This is a very powerful force, and we will see this later in the semester when we talk about fluid and electrolyte movement, and I can just preview it for you here. So if we have a container that holds water, and this water is pure water, no electrolytes, okay? This pure water into which we put a osmotic cell. So a cell that has the correct concentration of sodium and all those other things. Remember, we looked at that. Well, it's too far back for me to find. But, the 142 millimeters of mercury, 142 milliequivalents per liter of sodium. And we'll take that osmotic cell and drop it into this water. What's going to happen? Well, because of that huge gradient from the pure water to the saltwater solution, that water is going to enter into the cell rapidly, and it's going to cause this cell to explode, right? It's just going to explode. Guess what that pressure gradient is between a osmotic cell and pure water? It's 5,400 millimeters of mercury. That's huge. Can you imagine a blood pressure of 5,400? I don't think so. But those are the same units. So that is the force that the plasma membrane would have to endure in that crazy thought experiment. Okay, so that's osmosis. Remember, osmosis is net diffusion of water. Now, in addition to some other ways that we're going to talk about, there's an easy way for substances to get into a cell. And one of those ways is called endocytosis. And that's where we have the extracellular fluid here. So we have the ECF, and here we have the ICF, right? And so this extracellular fluid and substances in it want to get into the cell. Your cell can accommodate that through this process of endocytosis, where it just invaginates like this, breaks off that piece, and then that piece goes in here as a vesicle that can be dealt with at another time. There's something called receptor-mediated endocytosis. So how does the cell know that it's going to bring something into it? Well, it has to often be signaled to do so. And so one of the ways that signaling can happen is with a receptor here on the cell membrane. And that this is for the LDL receptors. So there's upregulation and downregulation of LDL receptors. Just as an example, when the LDL receptors get full, they get endocytosis. They undergo endocytosis, and they create these vesicles inside the cell then. And those vesicles then get attacked by the lysosome, which we talked about before. Remember, it contains the hydrolases. And this hydrolysis can happen now where it breaks down these cholesterol particles, and then that liberalizes free cholesterol for cell use. And then it can take that remnant receptor, that protein that's specialized to accept LDL particles, and it can repurpose it back to the cell membrane so that it can bring something back in. All right, that's receptor-mediated endocytosis. So again, in this case, it gets the LDL particle, brings it in through that endocytotic function, liberalizes the free cholesterol from it, and then repurposes this receptor back to the cell surface so that it can grab more LDL particles. There's other ways that transport of small molecules can happen. One of them is the transport of lipid-soluble molecules through simple diffusion. It could also be through an electrochemical gradient. There can be transport of water-soluble molecules that go through some transmembrane protein, and we'll talk about those in a bit. There can also be two types of protein transport: passive and active transport. Passive transport is just a type of diffusion through that membrane protein, and active transport requires some protein pump. And so we have a sodium-potassium pump, for instance, that allows us to move electrolytes around across that membrane, and there are others as well. The three main types of transporter proteins that we have are these ATP-driven pumps, and then we have carriers, which take the substance from outside the cell, goes into the carrier, and then the carrier gets an Uber for that substance to bring it into the cell. And the final type is this channel protein. Channel proteins are the simplest. It's just a hole, a pore in the membrane of the cell, and things can go through directly into or out of that cell. The types of active transport pumps that you may recognize are, again, the sodium-potassium pump. We have the calcium channels, right? Remember, calcium channel blockers block this membrane transport in smooth muscle cells. And then there's ABC transporters, which we'll talk about. Membrane proteins that I introduced earlier, remember, can be of these two types. They can be carrier proteins, where something comes in here, gets absorbed or gets attached to this carrier protein. Then the protein finds an Uber for it and brings it into the cell. Or we can have the ion channel, where potassium can just move freely through that channel. Now, these membrane proteins participate in diffusion and also in active transport. And the two specific types are shown here, the channel protein and the carrier proteins. Remember, the carrier protein grabs that substance, transports it down here, gets an Uber, and takes it directly into the cell. The channel proteins are just a pore that opens up that allows things to move in or out. So simple diffusion happens through the channel protein, and carrier proteins can be involved with active transport or facilitated diffusion. The sodium-potassium ion pump is pictured here, and again, this is a transporter protein that has binding sites on the outside. Remember, this is the extracellular side. So this is the outside of the cell, inside of the cell. And so that potassium binding site is here. With energy from ATP, it activates this carrier that brings the potassium in and sends sodium, which is bound here on the inside of the cell, out to the extracellular fluid. Again, this requires energy. That's a very important thing to remember. This doesn't just happen on its own. It requires energy to happen. The membrane calcium transporters are depicted here. Again, we need energy in order for this to occur. And then remember I said there's that ABC transporter, the ATP binding cassette transporter, where there's an ATP packet that gets connected here, opens up this channel and allows that substance, whatever it is, whether it's an ion, amino acid, a peptide, a sugar, or an other hydrophilic molecule, to go through the channel and into the cell. That's an ATP binding cassette transporter. Now there are passive transport carriers too, and a great example of that is the insulin receptor. So the insulin receptor exists on the cell membrane, and when insulin attaches to it, it activates this and sends a signal to these GLUT4, which is glucose transporters. Glucose transporter is equal to GLUT, right? That's where that comes from. So these GLUT transporters then allow the receptors for glucose on the cell membrane to open up and transport the glucose directly into the cell. So let's look now at the resting membrane potential. So this is that electrical charge when there is no net ion movement across this plasma membrane. And so largely it's the potassium ion that is responsible for this resting membrane potential. Remember that the majority of the potassium ions are intracellular, about 140 milliequivalents per liter intracellular concentration. And the extracellular concentration is in the neighborhood of 10 milliequivalents per liter. Okay? In the bloodstream, it's 3.5 to 5, right? 3.5 to 5.5 potassium concentration in the serum. So when the cell is in equilibrium, those charges, intracellular and extracellular, are about equal, right? They're about equal. And that is the resting membrane potential. Now, what changes that then is the movement of sodium. Now, in this case, you can see that having a normal serum potassium makes the action potential have this relationship to the threshold, right? The threshold is here. Remember, with that threshold comes an all or none response. So as the electrolytes move, once they get to this point, there's an all or none response. It goes up, the electrical charge changes, then it comes right down, and it goes below the threshold, at which point it can't elicit another response until repolarization happens. When the serum potassium is high, that changes this relationship so that now there's a much smaller difference between the end phase and the threshold. And so it's easier to cause mischief. It's easier for this all or none response to happen because there's less distance between here and here. When the serum potassium is low serum potassium, now there's a greater difference between the threshold and the resting membrane potential. That is now minus 100 millivolts instead of the usual minus 90. And so now it's harder to get to that point to cause the all or none response. So that's how differences occur in the resting membrane potentials. It's based on the concentration of whatever ion we're talking about. When we talk about muscle, particularly cardiac muscle, we're talking about potassium. So the thing then that triggers that membrane depolarization is sodium influx, and the sodium influx is stopped, and then potassium efflux, that is sending potassium out of the cell, begins to happen. So we stop the influx of sodium and we accelerate the efflux of potassium. When we look at myocardial action potentials in particular, and again, this action potential can happen for any number of cells in the body. It can be muscle cells, can be neurons, can be cardiac, it can be skeletal muscle, etc. Smooth muscle, whatever. So the action potential that we typically look at that we typically think about is the action potential of the myocardiocytes. And again, we have in phase 0, this is phase 0, we have influx of potassium in. That's until we hit that all or none response when the potassium starts to go out, right? Potassium efflux happens. Immediately thereafter, calcium comes back in, and we have more potassium going out before we go back to the beginning of sodium moving in. That's the action potential. That's the way that the heart works. It's a gradient between electrical charges on either side of the membrane. Now, how do cells communicate, and what is it that's responsible for signaling growth? Well, we have different cell signaling strategies in our cells. One of those cell signaling strategies is a function called the gap junction. This is where these channels exist between cells. So you have two cells sitting side by side. You have a cell here and a cell here. And then you have this gap junction, which is a connecting channel that helps to synchronize these cells. So for instance, cardiac myocytes. We're going to be talking about a lot about cardiac stuff, right? Because that's my background. So that's the thing that comes to mind when I'm describing most of this stuff. So these cardiac muscle contractions are synchronized in large part because of these gap junctions. But there's also direct cell-to-cell surface contact that can be involved in signaling. And this is an important thing in fetal development and also in immune responses. Okay. There are signaling molecules. We can also call them ligands. These ligands are substances that are picked up on the receptor side here that signal something. And so this ligand signaling goes from one cell across the the intercellular space, the extracellular space, to the target cell. And so that's one way that cells can exert an influence on another. They send a specific signal that is picked up by a receptor on the target cell. Another way that we can do this is with some carrier protein taking the signal to the cell and having it cross the cell membrane and go directly into the cell and have its effect. And then, of course, we have the gap junction that we just talked about. It's important that you learn this slide because throughout the semester, I'm going to be referring to paracrine function or autocrine function or hormonal functioning, and you're going to need to know what that means. I don't want to go back and force you to look these terms up every time, so I want you to learn this now. I'm going to give you examples of these and use them in our discussions throughout the semester. So, contact dependent is where you have one cell that has a substance on it, a messenger, that gets picked up by another cell. All right, so it's one cell that is expressing something to another cell. This is a large part of how our immune system works from this contact dependent stuff. Okay, so learn that, immune function. Paracrine function is where you have a secreting cell nearby a target cell, and when the substance is secreted, it's picked up by the target cell and some effect takes place. Autocrine functioning is where a cell has a signal that it sends out and is picked up by itself. So the cell has a signal that it releases, and that signal that it releases affects itself as well. Hormonal, obviously, is easy to remember because we're accustomed to this. Insulin may be a good example of this, where you have a hormone that's released that is insulin, and that insulin gets taken up on the target cell after it's been released from the pancreas, right? It goes from the pancreas through the bloodstream, goes to every cell of the body, gets picked up on the membrane of every cell in your body, and causes the insulin response. There's neurohormonal secretion. This is what we're going to talk about when we talk about the renin, angiotensin, aldosterone system. That's a neurohormonal thing. And then there's neurotransmitters, where we have a release of acetylcholine, for instance, that gets picked up by the postsynaptic receptor cell, and we have a increase in contractility on the other side. So, for instance, in the alpha adrenergic system, the alpha-1 receptor is postsynaptic. This is the alpha-1 receptor right here. It picks up this acetylcholine that's being released and causes vasoconstriction. Okay, that's the neurotransmitter method that we're referring to here. Okay. Next, we're going to talk about cell growth and regulation. This is an important topic because how do we regulate cellular growth? Why do some cells continue to reproduce, like cancer cells? Why do they continue to reproduce when other cells know to stop? Well, in order for cell growth to occur, normally there needs to be some signal. And these cell cycle controls are responsible for the birth and death of cells. Okay? There are effects that happen because of protein metabolism and receptors accepting growth factors, for instance. There are other growth there's other growth that happens because of neighboring cells telling it to do so. So, for instance, in the heart, if you have hypertension for a long time, there's a an increase in muscle mass called hypertrophy that happens because of that constant pressure overload. These neighboring cells release paracrine signals to one another that tell them to get bigger. And then there are other factors, other protein signals that tell us when the situation is ideal for replication. And finally, one thing I didn't include on here is apoptosis. There's a cell signal that will begin the process of apoptosis. Now, the cell growth happens and reproduction happens in this process called mitosis. And this is asexual reproduction, where the cell is told that it should reproduce. And so it goes through this cycle and splits itself into two identical components. Meiosis, on the other hand, is where the nucleus divides donates half of its self, half of the genetic material, to a new cell, and that combines with the other parent, and then you have an offspring. So that's sexual reproduction. Sexual, and this is asexual. Mitosis is cell reproduction. And is asexual. Sexual is birth of offspring, right? Offspring. Now, how does the cell know when to get ready to reproduce or divide? How does it know when to divide? How does it know when to stop? Well, those are all functions of this very complex thing called the cell cycle. It's important that you have some familiarity with this because a lot of chemotherapeutic agents are targeting specific phases of the cell cycle. So the 4 phases are gap 1, right, or G1. So this is where it starts. And then the S phase of DNA replication happens. Then the G2 phase happens here. Then we go into the M phase, which is mitosis or nuclear division. And then we go back to G0 and then G1. So this cycle is the cycle that is responsible for cellular reproduction. And it is regulated by signals, paracrine or autocrine or endocrine signals, and results in cell division. As I said, some of the chemotherapeutic agents target specific areas, particularly here in the G2 phase. And when we affect that G2 phase, we interfere with that reproduction so that it can't happen. And that was the old-fashioned way of developing cancer drugs. Of course, when we affect reproduction of abnormal cancer cells, we also affect reproduction of rapidly dividing cells. Those rapidly dividing cells are often endothelial cells, and that's why you have all of these terrible side effects like nausea and vomiting and stomatitis and things like that with chemotherapeutic agents. So the life cycle of the cell has two major parts to it, right? It has number one is the cell birth or replication, and number two is the signaling of cell death or apoptosis. One of the things that can stimulate apoptosis is cellular injury, but it doesn't have to be that the cell is told to die. With reversible cell injury, for instance, we can fix the cell, right? So the first thing that happens in reversible cell injury is this hydropic swelling, where the cell swells because of the accumulation of water. The first thing that happens in this cellular injury, number one, is failure of sodium potassium pump. Okay? That's the first thing that happens. And when that happens, you have increase of water going right in, right? Water just shoots right in to the cell. So let's take into account the idea of fatty liver. So that's what's pictured here. Fatty liver, can't write liver. Fatty liver is shown here, and that causes this accumulation of fat inside the liver cells. And that accumulation then makes it difficult for the cell to function normally. There may be an abnormal, so in a separate idea, there may be a protein mutation that has changed the way that these proteins fold and get transported inside the cell. We may have abnormal proteins that are being produced that accumulate in the cell. And this is what happens in PKU, for instance, phenylketonuria. There's an accumulation of an abnormal protein in the neurons that results in mental retardation. There may be an accumulation of substances due to a lack of an enzyme. And so what happens in cystic fibrosis is that there's an absence of an enzyme that transports chloride. And so chloride builds up inside the cells, and that's why you have thick secretions and mucus and things like that. And then you may have ingestion of indigestible materials that get into cells and clog up the works in here too. Okay? And so an idea an example of this would be black lung, where you have this coal dust that becomes intracellular and your cells can't get rid of it. There's no way to dispose of it, and it gums up the works in your lungs. So here is that example of silicosis. Here's an example of fatty liver on the left. So intracellular accumulations are another thing that can cause the beginning of the end for a cell. Now, before we get into talking about apoptosis and cellular death, we're going to talk a little bit about the adaptive cellular responses. This is another commonly recurring thing that we're going to see throughout the semester. So I want you to learn these now so that we don't have to revisit these definitions throughout the semester. So normal cells line up like this. They touch side to side. They have a basement membrane here that they adhere to, and everything is nice and orderly, right? It's orderly. If cells aren't used, then there may be atrophy, right? So somebody has a stroke, they have disuse of their left upper extremity, and so as a result, they have atrophy of those muscle cells. We talked about LVH, left ventricular hypertrophy, from hypertension or aortic stenosis or something. There's also hyperplasia. And by the way, hypertrophy is just where the cells increase in size. In hyperplasia, the cells increase in number. So you have a hyperplastic condition where the cells pile on and grow excessively. So maybe an example of this would be a callus, for instance, where you have, now that would be a normal thing to have happen. That's a normal response, but it's an adaptive cellular response because of constant pressure or friction or something like that. The things that we get concerned about are these two in particular. Metaplasia, which is conversion of one cell type to another. So for instance, the distal esophagus has columnar epithelial cells, pseudostratified epithelial cells, and then with repeated injury, the body tries to overcome the acid environment by recruiting goblet cells onto the scene, and those goblet cells secrete mucus. And that metaplasia that happens, where we replace one cell type with a different cell type, in this case, the stratified epithelial cells are replaced by goblet cells. That metaplasia is a precancerous lesion. And then, of course, dysplasia is disorderly growth, and that is cancer. Sorry, this is on the side of the page, and I have trouble holding my hand to write, so cancer. All right. Now we're going to talk about cell death. Now, there are two ways for cells to die. They can die a messy death, which is necrosis, or they can die a nice, neat death through apoptosis. So necrosis is where, for instance, there's just overwhelming injury to the cell. That causes swelling of the endoplasmic reticulum and the mitochondria, and cellular swelling because of failure of the sodium-potassium pump. Water rushes in, the cell expands, it explodes outward. The inflammatory process kicks in to pick up all the pieces, and that's the necrotic version of cell death. On the other hand, there's a nice, neat way where the cell is told that it's done, and it should pack its belongings neatly, and it makes all these nice little packages with the organelles and sends them out from the cell as the cell is fragmenting. And then phagocytosis picks up these apoptotic cell fragments and recycles them. It recycles those apoptotic cell fragments so that the body can use them for other things. Now, why is that important? Well, because, among other things, phosphate is a pretty rare substance on Earth. In fact, we're running out of phosphate, but we have about 100 years to worry about that, I'm told. But it's also rare for your body. So we need to conserve phosphate as much as we can, right? That is one reason that apoptosis is good, because we can take the phosphate from the ATP molecules, for instance, and we can repurpose them and use them elsewhere. So apoptosis is a nice, orderly process. No inflammatory response to be concerned with. We don't have pus formation. It's just neat. Everybody goes off into the sunset. Necrosis, which we started talking about, happens and can produce any number of these consequences. It can produce coagulative processes. That is what starts from ischemia, and then it ends with degradation of the plasma membrane. The plasma membrane breaks open, all the contents leak out, there's an inflammatory response, yada yada. There's liquefactive, where there's just the dissolution of dead cells, and liquefaction of the lysosomal enzymes. So we know that those lysosomal enzymes, the hydrolases, will dissolve stuff, right? Not a good thing for these lysosomes to be relegated into the extracellular environment and spill their hydrolases that will affect proteins, sugars, and lipids. We don't want to break those substances down in healthy cells. There can be fat necrosis, which is death of adipose tissue, and then there can be this thing called caseous necrosis, which tends to happen from tuberculosis disease in the lungs. Okay. Gangrene is cellular death in a large area that's from an interruption of blood supply. Okay. There's a couple different kinds. There's dry gangrene, which is that coagulative necrosis that we see blackened, dried, dead tissue that's demarcated from healthy tissue. We have wet gangrene, which is a form of liquefactive necrosis, and we see this typically in internal organs, specifically the gangrene of the pancreas can be fatal. And then gas gangrene results from infection of necrotic tissue by anaerobic bacteria like Clostridium, and that forms gas bubbles that damage muscle tissue, and this also can be fatal. So those are the types of gangrene that you should know about. Remember that necrosis occurs when the injury is too severe to recover. Okay? The cell can't adapt, and therefore the cells die. There's local and systematic indicators of death: pain, elevated enzymes, inflammation, etc. And then contrast that with apoptosis, or programmed cell death, where there's an intracellular cascade of events that lead to cellular suicide, essentially, and no inflammatory process. Okay, and there's two types of signals that lead to apoptosis. There's withdrawal of survival signs. So let me just explain this quick. We have cells that are told to stay alive in a number of ways. One is this matrix protein. So all these cells live with the matrix protein surrounding them, and as long as they're in touch with that, the cell signal is received that it should continue to be okay, right? A-okay, A+. Another thing that can happen is that you'll have signals from paracrine sources or endocrine sources, or whatever source is picked up that the cell responds to. And then the third thing is these gap junctions or other paracrine functions that this cell is telling this cell that it's part of the community and can still be alive and doesn't need to die. But when any of those signals are lost, whether it's the matrix protein, whether it's the ligands receiving signals from the endocrine or autocrine or other sources, or whether it's the side-to-side, cell-to-cell communication, one of those is lost, and that leads to breakdown of the cell and an apoptotic death. Fast ligand is an extracellular signal that binds to the cell and triggers death. So the fast ligand is the death signal. Oops. The death signal. When the fast ligand is received by the cell, it starts the process of cellular death by intracellular mechanisms, and that death then results in apoptosis. There are intrinsic pathways that trigger apoptosis as well. And so there is a protein in the body that's known as the, what do they call it? P53 is called the guardian. I'm just a terrible writer. Guardian of the genome. This guardian of the genome knows when too many mutations have happened in the cell and the cell should undergo apoptosis. When this protein is increased because of this DNA damage, it triggers the cell's own death. Okay? There is a class of cancer chemotherapeutic agents that target P53. I don't understand it. I heard about it recently. Somebody who was getting treated, it was a function of this P53 protein. So I know that it is a chemotherapy target. Perhaps you know about it and have had some experience with it, but I thought that was an interesting thing, that the guardian of the genome, which we've heretofore just talked about in pathophysiology and biology classes, is actually a target now for chemotherapy. And so these apoptotic intrinsic pathways can be activated that result in cellular death. How do cells get injured? Well, it can be because of any number of things. It can be because of ischemia, for one thing. So myocardial ischemia, a heart attack. CNS ischemia, stroke, right? Any number of any tissue can become ischemic due to loss of blood flow. And that hypoxemia, or the hypoxic injury that goes along with that, is the real harmful part. It causes power failure in the cell, destruction of the mitochondria, and an inability to produce ATP for powering the cell, such as the sodium-potassium pump. That's why the sodium-potassium pump is the first thing to go. It's the most common cause of cell injury, and can be detrimental, of course. When there's not enough blood flow getting to the tissue, or to the cell specifically, the cell doesn't have oxygen to run the metabolism. As a result, you get lactic acid production, and that develops, that makes the body acidotic. So initially, ischemic injuries are reversible, but after a certain point, when the mitochondria fail and the sodium-potassium pump fails, then the membranes become critically damaged, and it's you're unable to recover from that. When that happens, we get calcium overload in the cell, we have formation of free radicals, and the inflammatory process begins, and then the result, of course, is necrosis. Just a little bit I wanted to explain. I'd been in classes all the time where I heard about oxygen-free radicals. I had no idea what that was. Well, oxygen-free radicals, and you can read this chapter in Guyton and Hall, are specific forms of oxygen. So let me just explain. Oxygen can't oxidize into other chemicals by itself. O2 in that form cannot participate in the oxidative process. It first converts to an active oxygen form, some superoxide free radical like O2 with a negative charge, or, as we said before, H2O2, hydrogen peroxide. These are free radicals then that are normally cleared by the tissue enzymes, right? Like the peroxidases. Remember the peroxidase that we talked about? And those are catalyzed and detoxified. Sometimes an accumulation of these oxygen-free radicals causes problems. One example is with hyperbaric medicine. So the benefit of hyperbaric environments is that we increase the amount of oxygen that's available. That's a good thing to a point, but if you continue diving, right, if the pressure increases dramatically, then you get to a point where all you have is these oxygen-free radicals going around, and you can develop oxygen toxicity. So this oxygen toxicity happens because of these polyunsaturated fatty acids that are part of all the cell membranes become oxidized, and that causes cellular damage. So just a little note about oxygen-free radicals. I'm not going to ask you any questions about that, but if you ever wondered what the heck's an oxygen-free radical, well, it's this thing that's one of these charged particles of oxygen. Other mechanisms of injury, of course, we can have chemical injuries, like might happen with acid accumulation in the distal esophagus. We may have nutritional injuries that can be responsible for cellular demise. We may have infectious injuries, right? We talked about Clostridium that can have effects on healthy cells. And there, of course, can be physical and mechanical injuries that happen as well, ionizing radiation being one of them. Here's a great story from the New Yorker about a scientist at Los Alamos in, I forget when it was, maybe in the 1950s. He had this nuclear core and dropped a screwdriver into it. Saw a flash of blue light, and there was a surge of heat, and 9 days later he was dead. It's a fascinating, well, not fascinating, terribly interesting article about how ionizing radiation can result in cellular damage. So the two paths that it can take are free radical damage to cell structures, which causes acute cell destruction, or it can have DNA damage from that ionization that results in genetic mutations, potentially causing cancer, or the cell would recognize that those genetic mutations have happened and cause the cell to die an apoptotic death. Last thing for us to consider with the cells is this idea of cellular aging. Remember, these telomeres are at the end of the chromosomes, and every time that a cell replicates, that telomere gets shorter and shorter. And at some point, it will no longer replicate because the telomere has been expended. There are several theories about this, but this is really one of the areas of intense research among the people who study longevity. This cellular aging. We figure if we can stop cellular aging, then we may be able to slow down the aging process tremendously.